Hi everyone, it's Dr. LeBlanc with a video capture lecture on prokaryotic gene expression. Many genes in the cell are expressed at approximately the same level all the time. These are important genes like DNA polymerase and ribosomal proteins and ribosomal RNA. However, many genes in the cell are under more tight regulation. The regulation of gene expression from a gene to a functional protein takes place at three main levels by many different mechanisms. At the transcriptional level, at the translational level, and at the post-translational level. At the level of transcription, when you first transcribe a gene into RNA, genetic regulatory proteins can bind to the DNA and control the rate of transcription. In attenuation, transcription terminates soon after it has begun due to the formation of a transcriptional terminator. At the level of translation, messenger RNA that has already been produced can have repressor proteins that bind to it and prevent translation from starting. In addition, riboswitches, which are conformations of the RNA molecule itself, can prevent translation from starting by binding other small effector molecules. And finally, antisense RNA can hydrogen bond to the messenger RNA and prevent translation from starting. Finally, at the level of post-translational control, there are several different mechanisms that produce a functional protein. First of all, in feedback inhibition, the product of a metabolic pathway, what the pathway produces, can go back and inhibit the first enzyme in the pathway to downregulate the amount of product that's produced by the pathway. Finally, covalent modifications to alter a protein can also alter its function, such as adding phosphate groups or sugar groups or many other different kinds of moieties to a protein will change its function. They can either activate it or repress it or modulate it in one way or another. In subsequent videos, we're going to talk about two major operons in prokaryotes, the Lac Operon and the trip operon.